right. So today is going to be mostly structural, about DNA and chromosomes. And very early in the 19th century, people already knew that something must contain the gene in a cell. Okay. For many, almost infinite round of cell division, a cell will stay a cell, so something inside the cell must contain the information that make a cell a cell. Okay. But they didn't know what it was. They knew that cells contain many things, fatty acids, DNA, proteins. They knew all these things, but they did not know what is the genome made of. What kind of stuff? And very early they sort of say, okay, whatever it is, it must be contained in the chromosomes. And why did they kind of guess that? It is because they noticed that in the nucleus of a cell, usually a cell that is not dividing, just being there, doing what it does, inside the nucleus you notice a lot of stuff without really any noticeable organization. But just before dividing, they notice that suddenly something separates in the two future daughter cells. This is a very early event where something in the nucleus, something called the chromosomes, they condense and they separate and they go into the future daughter cells. Here the separation will happen and the future nuclei in the daughter cells will just be like the original nucleus in the mother cell. So, okay, this should be contained into the chromosomes. But the problem is that in the chromosomes, it's mainly two types of molecules, proteins and DNA. So say, okay, it's probably not fatty acid, it's probably not small molecules, it's probably protein and DNA. And as you know, well, in 1953, Watson and Crick, they discovered the structure of the DNA. Before that, they knew what DNA was made of. They knew DNA was extremely simple, only made of 4 different building blocks. And they sort of said, that can't be, it's not possible for it to be the genome because it's just too simple. So they thought in the beginning that it was actually protein. Protein, yes, protein is making our cell work and enzymes and all these things. So maybe protein must also somehow code for our genome. So people didn't know. And it's until mainly 2-3 experiments that really say, you are wrong, this is not protein, this is really DNA. We know it's very simple, but still DNA seems to be what makes us us. And the first experiment is, as you can see, very early in the 1920s. This is by a scientist called Griffith. And very nicely what he did, he was using rat or mice, and he was injecting them with a living S strain of Streptococcus pneumoniae. So it's something that will actually cause lung infection in the mouse. And when you do that, unfortunately, the mouse dies of infection. But he had another strain of this Streptococcus pneumoniae, and this is an R strain. And this strain, even though this is Streptococcus, it is not infectious. You inject it in the mouse, you will have some multiplication of these Streptococcus, but essentially they will not cause disease. And the mouse will live. what they also did was, okay, let's kill these infectious strain of Streptococcus and let's see what happens. So they killed these bacteria. They killed them with heat, with a flame, very simply, and then injected these heat-killed S strain into the mouse. And then, as you would expect, the mouse just completely is fine. Okay, it doesn't die because you have killed all the bacteria. However, if you do the same thing, but you co-inject these heat-killed S strain with this living, non-infectious R strain, the mouse dies. The mouse dies. And if you actually collect, shortly after the mouse dies, if you actually collect blood and see what kind of bacteria the mouse contains, you actually see that somehow you are having S strain Streptococcus into the blood. Even though all the S strains were killed there, you actually extract the living S strain. So somehow something happened between these two strains that made the S strain recover. Okay. So that was a very, very nice experiment showing that somehow these two things, these two bacteria strains, exchanged something. And the living R strain somehow became like the S strain. So they must have exchanged something. So slightly later, well, about 20 years later, this took a long time because they had to establish method to do this, but they did exactly the same experiment. This is Avery and MacLeod experiment. They did the same experiment, but they didn't just inject like this. They first killed, and then they extracted different components from these infectious Streptococcus. And then what they did is they first separated RNA, protein, DNA, lipids, and carbohydrate. And then they mixed them up together with an S strain, with a living S strain, beg your pardon. And they see what has the ability of transforming the R strain. And then if you mix R strain with S RNA, nothing happened. If you mix R strain with S protein, nothing happened. Lipids and carbohydrates, nothing happened either. But if you mix the R strain DNA with the S strain that is killed, that is if you mix this living R strain with the DNA from S strain, then what you get is a living S strain that is able to kill the mouse. So this experiment showed that DNA is what transformed the R strain into an infectious S strain. This is quite a conclusive experiment, but people who still believe somehow that proteins was the genome, was the genetic information. They said, well, but you don't really know if you have removed all the proteins from your DNA. Yes, you have enriched in DNA, but have you removed all the proteins? You don't know. So they criticize the experiment quite a lot. And they say maybe what you are doing, this involves a lot of preparation, right? So scientists were saying, maybe you are actually doing something to the R strain. You are doing something you are mutating the R strain. Maybe there is a chemical that you are using that is mutating the R strain. And whatever you do here is not relevant. It's one of your chemicals that you are using that is actually transforming the R strain into an S strain. So essentially, they weren't at all convinced by this experiment. And it's until two other people who didn't use these Streptococcus. They actually used E. coli, Escherichia coli, this is dijoking. And this bacteria is very nice, it's not infectious in normal condition. But it can be killed by a virus called the T2 virus. And this virus, what is really nice, it's made of two things. As many viruses, it's only made of proteins, which is the component of the viral envelope, okay, and DNA, which we know now the virus will inject into the bacteria, and then the bacteria will use the DNA to make many, many viruses, and the bacteria will explode, and you will have many new viruses that will then go into the medium and infect other. So this is what they use, and they say, okay, the virus is made of protein or DNA, and we know the virus is using the bacteria to make more copies of itself. So this must be because the virus is injecting either protein or DNA. So let's check what the virus is doing. And very nicely, I think, in a very simple way, they prepared two types of viruses. The T2 viruses, but one population of T2 viruses, they actually labeled them with 32P. It's a radioactive isotope of phosphorus. And they, on the other hand, they had viruses that were labeled with 35S, which is also a radioactive isotope of sulfur. So what will happen in this case? What will happen in this case? Well, you can see it's written there. If you label the viruses with 32P, DNA will be labeled. Okay. DNA will be labeled, but if you label the viruses with S35, with 35S, proteins will be labeled. Okay. Because phosphorus is one of the main constituents of DNA, while 35S is one of the main constituents. Not really a main constituent, but it's really mainly contained in protein. So then you have viruses that are either labeled with 32P or either labeled with 35S. So what you do is that you infect the Escherichia coli with these two viruses. You wait until the virus interacts with the E. coli and infect them and inject whatever they inject. And then you just put this preparation in a blender. Just normal, not really normal, but it's just a blender that's similar to the one you use in the kitchen. And the blade will actually separate the viruses from the bacteria. So you will remove the head of the virus. You will remove the head of the virus from the bacteria. And then you purify via centrifugation the heavy bacteria that were infected by the virus. And then what do you get? What do you see is that these bacteria, they contain inside their cytoplasm, they contain 32P. Okay. They contain 32P, so they are radioactively labeled with DNA from the virus. Okay. So this shows that the virus is injecting DNA into the bacteria. And even if you check the next generation of bacteria that divide from these bacteria, they are still labeled with 32P. So this shows without ambiguity that really DNA is what the virus are injecting the bacteria. This is the genetic information of the viruses, and therefore is also the genetic information of all the organisms, because all cells use DNA as genetic information. Okay, so this is Hershey and Chase experiment, 1952. And as you can see, 1952, it's maybe in the same time as when Watson and Crick discovered the structure of DNA. So at that time, it went very, very quickly. And since 1950s, the molecular biology has made a huge advance, and Many things have been discovered. But really, we had to know what is genomic information, what is genetic information, and what is the structure of DNA. And then from these discoveries, then it went extremely fast. All right, but let's see a little bit about the structure of DNA, which you know. Structure of DNA is just like this, okay? Two anti-parallel strands, because the 3' knob and the 5' knobs are different. The 5' knobs contain a phosphate here, and they are anti-parallel because the 5' end is on the other side. Okay? And very nicely, what Watson and Crick found out is that this double strand of DNA is actually associated. Mainly here, this is hydrogen bonds between C and G and between AT. Okay? And it's not just like a ladder like this, it sort of curves into a double helix. And what is really great with this is that it directly gives a hint as to how organisms can actually inherit from the DNA of their sire. For example, you can see that since one strand of DNA is the complementary strand of its of the other strand, then it's very nice because this is a structure that is perfect for giving information to the next generation. Okay, it's complementary. Therefore, the rules of inheritance were transparent from this structure of DNA. And well, this is a little bit more details. You already know this thing. I won't go into it, but maybe let's just say that 1 turn of the double helix is about 10 nucleotides. And each nucleotide, each pair, this is called a base pair. Okay, this is a base pair, this is another base pair, and always you have adenine with thymine and guanine with cytosine. And then inside the double helix, all these base pairs are stacked almost completely parallel. Okay, and this structure, this double helix, is necessary for DNA to be stable. Okay. And it's not completely random. This organization defines specific structure because you have a minor groove here and you have a major groove. As you can see from the previous here, this is the major groove here and this is the minor, okay, which has different atoms sticking out. Okay, and this is important for the interaction of DNA with protein. Okay. And as I said, the structure of DNA provides a mechanism for heredity. Well, you have this in the book. This is not so informative, but the long string of nucleotide, somehow, when scientists discovered this structure, they sort of said, okay, what we are having here, it's a long string of nucleotides, so this must be a code. That if we're able to decipher this code, then we will be able to know that maybe this code directly codes for the amino acids that are present in protein. Because scientists knew that proteins are doing everything in the cell, so then the DNA must code for the sequence of amino acids. But that was not evident from the DNA structure. If you look at the DNA structure, this double helix, it's not evident what's the code. Okay? So then more experiments had to be done to really know how this sequence of nucleotides code for the protein. Okay, but if you see here, let's imagine a theoretical double strand of DNA that actually codes for three genes, gene A, B, C, each coding first for its RNA, which is a direct copy of DNA, and the RNA being translated into protein A, B, C. So this is very easy. But what is important, I think, in this chapter is to realize if you take one of your cell, maybe like what, 10, 20, 50 micrometer, and if you are to extract the DNA from your cell, you have about 2 meters of DNA inside your cell. 2 meters. So that's about, you have a string of like 40 kilometers. And I'm asking you, can you put this string of 40 kilometers into a tennis ball? That's the same thing. In terms of dimension, that's the same thing. In a small cell, you have 2 meters of DNA. So how is that achieved by the cell? This is the purpose of this chapter. Well, one of the purposes of this chapter. If you can extract all the chromosomes of a dividing cell, so it's a cell that is actively dividing, okay? Which means it's a cell in which the chromosomes have been condensed like this. And there is a way by which you can specifically label each chromosome. Okay? So what you have here, it's all the chromosomes from one single cell, and each chromosome has been labeled. And what you notice is that you have, of course, pairs of chromosomes. So you have the pair of chromosome 1, chromosome 2, 3, 4, and then you have X and Y. All right? So you have 23 or 24 different chromosomes in your cell. How can you label these like that? Can you think of a way by which you could actually achieve such chromosome painting? Each chromosome is labeled with a different color, and you can see chromosome 1, there are 2 chromosome 1, there are 2 copies, so of course both copies are yellow. Can you imagine a way by which you can do that? How could you achieve? This is not fake. Okay? Each chromosome really has been labeled with a different fluorescent dye. But how can you do that? It's getting the end of the year. You're all sleepy. How could you do that? Thinking about what chromosomes are. I told you earlier they contain DNA and protein, right? So how could you think of a technique by which you could specifically label each chromosome with a different color? So we have a funny color here. We have a funny, this is a little bit like brownish thing, but there is a little bit blue there. But that's because if you look in the original, if you look at this chromosome inside the cells, you can see that this chromosome overlapped with this one. So then they are having a mix of color. Because they just cut optically these and then align them. So this is why you have a funny color. So how can you do? Think about what it contains. You could use radioactive isotope. But I don't want to get close to it. Theoretically, you could do, but the problem is that how do you differentiate? If you use a film that reacts to emission, alpha, beta, whatever, then how do you differentiate different emissions? So it's possible, but then all of them will have the same color, black and white. Here we are having different colors. Different colors. This is fluorescent, right? So they have different colors. It means that they are labels that have a different spectrum of emission. Okay? So this label here, when it's excited by light, probably with a different excitation spectrum, it's giving you light of a different wavelength, right? So the color is different. The fact that you are using different labels is great, but how do you make this label stick to each different chromosome? Maybe, I don't know, maybe this label here is the same as this one, but still. And maybe the red one is also the same. So you can use, you don't really need to use exactly 23 or 24 different labels, but you know, if you have maybe 10 or 12, you can do that. But how do you make them stick? You cannot just have fluorescent labels and they will go everywhere. You will have a mix of strange color. But here, one color, one chromosome. So, thinking about DNA, this is made of DNA and protein, so. I'm not saying. Mainly there are two possibilities. There are two possibilities. One is not so good, the other one is the one that they actually do. So, how could you paint each chromosome with different color?
What are the building blocks of chromosomes?
Hmm?
Building, what are chromosomes?
It's made of protein and DNA. This is only what you have: protein or DNA. So there are two possibilities, right? The two possibilities I just mentioned. It has to do with these two, either protein or DNA. So. Imagine the structure of DNA. What is DNA? Imagine the structure of protein. How could you? So essentially the question I'm asking you is either you need to find something that interacts with protein or something that interacts with DNA in a chromosome-specific manner, right? This is a different way, more precise way to ask the question, but it gives a lot of hint. So how could you do that? Let's first focus on protein. If you want to label the proteins that interact with DNA in the chromosomes, how could you make them fluorescent? What kind of tag can you use to label protein? From a previous chapter. From a previous chapter. Actually from just previous one. About proteins. something that binds to protein, to whatever protein you want. What could you use? I'll have more reaction giving a lecture to the terracotta army in China. Come on, anybody, don't, if you know, don't wait anybody else to say the answer. If you know, just say the answer.
We may use antibody, but I can't think of how to use it.
Yeah, exactly. So you can use an antibody if you find a protein that interacts with DNA in chromosome. Then you could have an antibody that is labeled with a fluorescent tag. So you have maybe 10 different antibodies or maybe 20 different antibodies that each recognize proteins of each chromosomes. So the protein will interact with its cognate protein, and they are labeled with different fluorescent tags, so this will work. Okay. The problem is using protein is there is no protein that are specific to each chromosome. So if you do that, yes, it's possible. You can have an antibody that is tagged with a fluorescent protein that will interact with chromosomes, but all chromosomes will be the same color. Because the protein that interact with the antibody, the protein that interact with DNA are the same in all chromosomes.
Yes.
Right? So it will not work. Well, you can do the same thing, but they will all be green, or they will all be red, or whatever. Or they will all be multicolored. So it's not so good. So for that, the next option is better. You want to get these tags to interact with DNA. Because DNA, it's really specific. The sequence of chromosome 1 and the sequence of chromosome 2, 3, 4, the sequence of chromosome X and Y, they're all different. Right? So what could you do? You cannot really make an antibody that interacts, that recognizes a specific sequence of DNA. That's not so feasible. So what could you do? So protein is out. Protein is not good. It's not specific enough. So what do you do?
RNA?
You could use RNA. You could use RNA, but RNA is not so stable. RNA is not so stable. But yeah, you're going the right way. You're going the right way. You can actually use DNA. You can use DNA that have the same sequence, not the whole thing, but you can use DNA, small bits of DNA that have sequence that is specific to each of the chromosomes. Maybe 100 nucleotides would be enough, because 100 nucleotide string would probably be specific to each chromosome, and this bit of DNA is labeled to a fluorescent, it's bound to a fluorescent label. So when you mix the chromosome together, you can actually have interaction of the single strand DNA into the chromosome. This is possible. This is possible. So they are labeling these chromosomes with short bits of DNA that has the same sequence as some part of the chromosome. This is what they are doing.
I have two questions.
Yeah, I like this one.
First is, how can we make DNA brighten? And second is, DNA is usually double-stranded, so can we attach DNA to double-stranded DNA?
That's two good questions. So the first question, simply, if you are having, let's say, chromosome 1 here, you are having chromosome 1 here, and how do you make DNA bright? You cannot make this DNA bright, but what you can do is maybe you are using a probe. It's called a probe that is complementary to one of the strands of DNA, and in addition, it has a tag, any kind of molecule that is fluorescent. So the only thing you need to do is to hybridize this tag, this probe to your chromosome, and then you give light, UV or whatever. This fluorescent tag will get excited and will emit light. So then the chromosome will actually be like this. The second thing is that how can you actually have this, because DNA is double-stranded? Well, actually, there are different ways. You can actually have triple helix. This sometimes happens. If you bring one single strand DNA, it can actually hybridize to a double helix. This is possible. It doesn't really happen in cells, although some people have said that they have observed a triple helix. Okay. But certainly, when you put a high amount of these probes, they can actually recognize the sequence, even though it's already double-stranded. They can insert inside the double helix. But what you can also do is to heat. You heat a little bit these mix of chromosomes, and then you bring your probe, so that heating it will sort of partly separate the chromosome, the two strands of each chromosome, so that your probe, which are small, will actually insert themselves into it and form. So you can use both ways. And of course, you can imagine that they are just not using one single probe for each chromosome. They are probably using probes that will hybridize to each chromosome, not only at one location, because otherwise you would only have this part maybe to be fluorescent, or this one, but they are using probes that are probably overlapping and can go everywhere in each chromosome. Okay.
One more question, okay?
One more question, okay.
Is more feasible than using many antibody, because many short strand with bright protein is difficult, I think.
This is not protein.
Oh.
This is not protein. It's fluorescent molecules, but it's not huge protein like GFP or red fluorescent protein. These are small tags that can actually give off fluorescent light. So then it's much easier. Right. But there is another way. You can do the same thing, and this is actually relatively recent. But before, what people were doing is that they were doing the same thing, but with probes that can mainly differentiate between AT-rich region and GC-rich region. And in this case, all the chromosomes were gray, like in a radioactive tagging. But the banding was different. So you have here, for example, you have chromosome 12 here. This chromosome 12 is normal. And you can see there is a banding pattern which corresponds to the alternation of AT-rich and GC-rich region. And this banding pattern is specific to each chromosome. By just looking at the pattern, you can recognize which chromosome it is. Okay? And this is also via these, like a probe that can recognize AT and GC-rich region. And what it was used before, and still sometimes used nowadays, is that thanks to this pattern, you can actually see when people have some genetic disease, because you have, for example, this one, you can see that the banding pattern is that of chromosome 12, but there is this one in addition, which corresponds to chromosome 4. And this is one of the chromosome abnormality that you have in some diseases. Okay. This picture, of all the chromosomes in the cell is called a karyotype. You actually take a picture of all the chromosomes to see if you have any abnormality or see the structure of chromosome. It's called a karyotype. Okay. Chromosomes contain long strings of genes. So this is only maybe 0.5% of the DNA of the yeast genome. And then you have, of course, two strands, 5 to 3 prime and 5 to 3 prime. And what you can see here is that genes can be on either strand. Okay? Gene can be on the plus strand, or gene can be on the minus strand. It doesn't matter. Okay? Some genes can really be overlapping. In this case, genes are not overlapping. So the two genes here, there are heterogation where there is no gene on the minus strand, but this can happen. You can have overlapping gene in antisense to each other. This can happen. Okay. And you can have very long genes, like this one, for example, and very short genes, let's say like this one. Okay, so genes can be just about anyone. And as I told you before in the beginning of this lecture series, there is no simple relationship between the total length of your genome and the amount of genes you have and how advanced you are in evolution. It's a very, very complex relationship. And for example, if you compare two muntjac, like you have this Chinese one and you have the Indian one here, you can see this is a little bit different, but essentially it's the same animal. They can breed together. But if you actually take a look at their karyotype, it's completely different. Even though the sequence of their gene would be like maybe 95% identical, you can see that how their genome is organized in chromosomes is completely different. What you see here, these are the mitotic chromosomes, so it's in a cell that is dividing, and then you can see that this Chinese muntjac has many chromosomes, and this one has only like 1, 2, 3, 4, 5, or something. So it's completely different. Although the sequence will be almost identical, how they are organized in the genome is completely different. So again, you cannot really use how many chromosomes you have to know how close you are to other species. Specialized DNA sequences are required for DNA replication and chromosome segregation. So first of all, it's more complex than that, but let's just say that cell division is made of two phases. There is the interphase, during which the cell is just waiting, or during which the cell is synthesizing its DNA. And synthesizing its DNA, replicating its DNA, it's more precise. So it's replicating each strand of DNA. So you have this chromosome here, and you have a second chromosome here. During the interphase, DNA synthesis will occur so that at the end of the interphase, each chromosome will actually exist in two copies. Okay? Like this one chromosome is duplicated. So that during the mitotic phase, the M phase, what happens is that each copy of chromosome will be pulled into each daughter cells by something called the mitotic spindle. Okay? So each chromosome, each copy of chromosome will be pulled into the daughter cells. So remember this interphase and M phase. All the chromosomes we have seen so far, this one and this one, they are all from cells in the M phase. Okay? And they have this duplicated chromosome. Okay, but as you can see, this will not happen just like it. Okay? These chromosomes being pulled apart from each other, this does not happen easily. So what you have here, imagine this is your double helix of a chromosome. Okay? And just it's not really drawn up to scale because you can still see the double helix here. But essentially, one chromosome is made of several parts. First of all, the end of each chromosome is called the telomere. Telomere is really the end of the chromosome, and it's very important for something we will see later. Then there is the replication origin. The replication origin is very important because it allows the two strands to be pulled apart and DNA synthesis to occur. You cannot synthesize DNA until you pull the two strands. Okay. So this is what the replication origin is. It's a specific region that allows some proteins in the cell to pull the two strands apart and start DNA duplication as you have here. And you also have the centromere, and the centromere is also very important because this is what interacts with the mitotic spindle. And because of that, you will be able to pull each chromosome into the future daughter cells that will happen in the interphase. Okay? And of course, in the interphase chromosome, you can see the chromosome is really unpacked. But during the M phase, as you can see, the chromosome is condensed into something that is much smaller. We will see about this one later. So please remember, telomere, this is the end of the chromosome. Replication origin, it's important for the synthesis to start to initiate the synthesis of DNA. And centromere, it's important for pulling the chromosome apart during the M phase. Something important, you can see two replication origins were drawn. Why do you think, especially our chromosome, it's not the case for bacteria. Maybe bacteria, they have one replication of origin, but each of our chromosomes maybe have many hundreds of replication origin. Can you imagine why that would be so? Why a bacteria with one single chromosome that is usually circular, and as you can see, bacteria genome is much smaller. Bacteria genome is much smaller than human genome, right? So why would bacteria chromosome, the name is the same, but the structure is a little bit different, but why bacteria chromosome would have one replication origin? And why our chromosomes would have more than 100 or so? Can you imagine why? Can you imagine why? This replication origin. Why would we each our chromosome would have 100, 200, 300 or even more replication origin?
Does the reaction happen faster or?
Yeah, this is exactly. Because it will be faster. If you have one replication origin, you will need to synthesize the whole thing at only once, and then it takes a long time. But if you have many replication origin, then very quickly new synthesis of DNA can happen much more quickly. So this is exactly to make it quicker. And bacteria, since it's very small anyway, it's quick and doesn't need many. Okay. So this is really scanning electron micrographs of a mitotic chromosome, where we can clearly see the centromere here and each chromatid of the duplicated chromosome. Okay. And you can see this is DNA, so it's really incredibly packed. Okay. Interphase chromosomes are not randomly distributed within the nucleus. If you actually take a look at a cell and you do the same thing that you did before, where you actually label each chromosome with a specific fluorescent tag, you can see that each chromosome actually occupies different region into the nucleus. You can see the chromosome 9 here, for example. And we do not understand exactly how this happened. It is not random, and it is changing. In different cells, the organization will be different depending on the genes that are expressed. But one feature of the nucleus that we can see simply by looking in the normal microscope is this one. This is an electron microscope, but this is the nucleus. So this is the nucleus here, with its envelope, double membrane. Okay? And inside the nucleus, you have the nucleus. And what is very interesting is that the nucleus is where you make ribosomal array. You have genes that code for ribosomal array. Genes normally code for protein. What we think of genes is coding for protein. But you also have ribosomes. And ribosomes, they are RNA, so they must... But ribosomes, they also need to be made. And they are genes coding for ribosomes. And very interestingly, all the genes coding for ribosome, they will actually cluster in one region of the nucleus. I'm sorry about this. This is very annoying. Maybe I should. There. Somebody is working. This is great. Somebody in my team is working and doing many things in the Dropbox, but that's annoying. I'm going to close this exit. Okay. All right. So the nucleus is not anything that is separated by membrane. It's only where all the genes coding for ribosomal RNA cluster. So there must be a lot of organization of all these chromosomes to be able to do such a feat. Because of course the genes that code for ribosomal RNA will be on different chromosomes. Okay? So somehow maybe you have a chromosome here that will sort of fold and go here where you have the ribosomal RNA gene. And then you have another chromosome who will be here, and then it will go inside here where you have a gene coding for it. So it's really, really kind of amazing. And we don't really know how the nucleus can be organized. And in addition to that, you have in the nucleus regions where you have dense sections, and you have sections that are quite bright. And these corresponds to different packaging intensity of the DNA. So where you have these dark spots, usually around the nuclear envelope, this is where you have heterochromatin. And we will see what later is heterochromatin. So whenever it's dense, it means that the DNA is pretty heavily packed, and usually genes are not so transcribed. And where you have A zone that is brighter means that it's less packed and it's more transcriptionally active. Of course, nucleolysis is a bit different in this case. Okay, the DNA in chromosome is always highly condensed. If you take a cell, you take the nucleus of a cell and you break the nucleus, what you get is this. It's like a spider web. It's all the DNA that is exploding out of the nucleus. And now, for example, the chromosome 22, it's 48 million nucleotide pairs. This is about 1.5 centimeter in length, if you really measure the length of the double helix, right? But during the mitosis, it measures 2 micrometers. So that's a compaction of about 10,000 times. This is incredible level of condensation. And during the interphase, when the chromosomes are not condensed like this, it's less compacted, but 20 times less than during mitosis. So still, you have a high level of condensation there. Okay? And because the level of condensation is so high, this is why when you break the nucleus, suddenly all this tension releases and DNA explodes. And if you have a population of cells, and for experiments, sometimes we need to break cells open. You can really see this DNA coming out when you break your cells open. Let's say you have in a small dish, you have like millions of cells. If you break your cells without breaking the nuclei, the mix will just be like water. The cell will be broken, but it will be very runny, just like water. But if you break the nuclei, suddenly it will become neba-neba. It will become very viscous because all the DNA suddenly is released into the solution. And it makes a very almost gelatinous mix because DNA is so long that actually when it loses its structural integrity, it's sort of unpacked by itself. So this is a pretty amazing thing there. But still we don't really understand completely how DNA can be packed in such a small, 1,000 times, 10,000 times more compact than if you just measure the length of DNA. Nucleosomes are the basic units of eukaryotic chromosome structure. Okay, let's see how chromosomes are organized. So let's say you do that, and you take a look at this. Okay, you take a look at one single strand there. What you see is this. Okay. Don't really see much what it is. It's called a chromatin fiber. So inside there you have DNA and protein. DNA and protein. It's called chromatin fiber. But let's say you add a bit more salt, you add some sort of soap or whatever, and suddenly what you see is that this starts to decondense, and what you see is like pearl on a string pattern, where you have a bead, a section, a bead, a section, a bead, a section. And what you see here is really a series of nucleosomes. A nucleosome, this is it. So what we see here, this is this one. So this is DNA, the double helix of DNA, that is wrapped 2 times around chromatin. And you can see you have one here, linker DNA, another linker DNA, another. So that the linker DNA and its adjacent core histones is called a nucleosome. So here you have many, 1 nucleosome, 2, 3, 4, and so on. And this is the basic structure of chromatin. And what you can do is you use DNAs, like it's a nuclease that will digest DNA, and it will only digest DNA that is free. So for example, when you have DNA that is interacting with the core histone, it will not be degraded, but by doing this, you will be able to know exactly how many turns the DNA will do around the histone. And it's about 1.7 turns. And one histone like this, one core particle, is about 11 nanometer. And then what you can do is further dissociate the histone from the DNA wrapped around the histone. And then you can see what the histone is made of. It's mainly made of 1, 2, 3, 4 sorry, I forgot that I can do that. It's not a good definite. It's an octamer, it's written here. So it's an octamer, which means that they actually, the whole thing is a huge complex of protein, but it's made of 2 dimers of H2A, 2 dimers of H2B, 2 dimers of H3, and 2 dimers of H4. It's not the only thing. This is very simplified. We will see more details later. Okay? So it's a multimeric protein. But in addition, what you have here, you do not see that there is something very important. So you can see here, this is the DNA, and you have each of the, I forgot the word. Can you please tell me? Not octamer or dimer, but how do you call one subunit? That's it. I forgot the word subunit. Sorry. I had forgotten the word subunit. I'm sorry about it. So you yeah, subunit. You know how bad can it be? So you have each subunit of the histone octamer, but in addition, for each of the subunits, you have here. We only see this one for each histone H3. We can see a tail sticking out of the core particle here, and the tail is extremely important. And you have a tail for the H3, you have a tail for the H2A, you have a tail for the H2B, and it's really critical for how chromatin is arranged and packaged. So we can play a film in which you have a good image of what is happening. So you have just here the double helix. And then on this double helix will come each subunit by subunit. And everything organized by a non-covalent bond. Okay. And we can see very nicely the tail of each of the histone subunits. But then it's more than that. Then these okay, so what we saw here very nicely is how this comes into this one. So there is after this one, there is a further level of compaction to become a chromatin fiber. Inside the cell, you don't have that. You only have that if it's kind of artificially unpacked. In the cells, you have chromatin fibers. Okay? Chromosome packing occurs on multiple levels. The last bit of the film, we saw that all these nucleosomes suddenly packing densely around each other to make the chromatin fiber. Okay? And how does this happen? Is because there is another histone, which is called the histone H1. A more common name for this one is the linker histone. And that what it will do, it will associate with the core particle and the linker DNA to change the direction of the DNA going out of the core particle. And it will make really each of the nucleosomes really tightly packed with each other. Okay? So this is another very important component of the chromatin. This is the histone H1. So what we have here then, and it gives you a nice idea of the level of condensation that this is doing. So you have a short region of the double helix, 2 nanometer. Then you have the beads on a string form of chromatin where you have the nucleosome here. This is 11 nanometer. Then you have the chromatin fiber of packed nucleosome. Here, the linker histone, we don't see it, but we can imagine that the linker histone is here and make sure that, you know, each of these nucleosomes are tightly packed. This is 30 nanometer. And then you have a chromatin fiber that is folded into loops. And still, there are many more levels to go to the mitotic chromosome. And we do not at all understand how this happens. How can this very quickly, within a few minutes, I mean, this mitotic phase really takes maybe 10 minutes, 15 minutes, sometimes a bit longer. So it's extremely fast. And how this happens, we have no idea. We know some of the proteins that are necessary for that, but we have no idea how this happens. And every time the cells divide, they must do it. Okay? And after division, they must then unpack the whole thing. So this is really, really an amazing feat of structural organization. So you can see here, net result, each DNA molecule has been packaged into a mitotic chromosome that is 10,000 times fold shorter than its fully extended length. So what we can see here, this is again electron microscopy picture of the two chromatin. And this is the end of one chromatin, and this is the end of the other chromatin. The regulation of chromosome structure. Changes in nucleosome structure allow access to DNA. Yes, because you can see this is the state in the interphase cell, this is the state of chromatin. Okay? But still, DNA, genes must be transcribed. DNA must be copied into RNA. Can you tell me how RNA polymerase can go inside and start to pull apart the two strands of DNA to make RNA? This is incredible. So we are going to see how this happens. Well, very briefly. You have non-histone proteins that can associate with histones, and it's called ATP-dependent chromatin remodeling complex. What they can do is actually make each co-histone, if you want, spin like this. So here, before this chromatin remodeling complex work, you have bits of DNA here that is wrapped around the histone and leaves here. But let's say you want to have access to this part of the DNA double helix. So what this chromatin remodeling complex do is simply winding the core histone so that more of the DNA here can be made available. And of course, it's not only one. They all kind of they will all be orchestrated so that when you have this chromatin remodeling complex, they can really decondense the chromatin to a dramatic level to make the DNA available for duplication, for gene expression, whatever. And of course, you can imagine these changes in the chromatin structure requires ATP. Okay, so these are the chromatin remodeling complex. And they are really linked to these histone tail. So you may have the question of how does the chromatin remodeling complex know where to go? Okay, I need to sort of free this bit of DNA. I need to work on this histone because the cell wants to have access to this bit of DNA. How do they know? And this is because of this histone tail. The histone tails are sticking out. Okay, the histone tails are sticking out outside of the core particle. And these tails are very rich in lysine, arginine, serine, and these can be modified by many, many things. For example, here what you have is M, it's called methyl. You have an acetyl here, and you have a phosphate. So these histone tails can be modified by phosphorylation, methylation, acetylation, and each of these will be a message for the cells to say, hey, you should bring your histone remodeling complex because something tells me that the cell wants to access this gene. Okay? And the meaning of each of these modifications is different. For each histone tail, each position is different. For example, this is a few. The H3 histone modification state. So if you have H3K9M, this is how we name these things. So this is H3K9M. What it means is, please make at this location makes heterochromatin, which means condense the chromatin. We don't need this, we need to silence this gene. So make heterochromatin. If you have H3K4M, H3K9 acetyl, this is a completely different message. This means that gene expression needs to be activated. And we have a third case, we have a phosphorylation coupled with an acetyl. This is also means please activate gene expression. So it's a really extremely complex level of regulation. But this doesn't explain. Okay. What I told you is that how do these complex remodeling, chromatin remodeling complexes, how do they know where to go remodeling? And then I tell you, they know because each of these histones is having a message on its tail to recruit these chromatin remodeling complex. It's not explaining anything what I'm doing. It's just one step. Okay, we have each histone has these things. But then what is the question you want to ask me?
How do these enzymes which phosphorylate or acetylate these tail know hey, this gene activate?
Exactly. How do these occur? How do the enzymes that actually add the acetyl, how do the enzymes that add the methyl, how do the kinase that add the phosphate, how do they know that that histone need to be acetylated? How do they know that? Okay. Because in the beginning, all of these are not modified. These come later, and these modifications can be added, but they can be erased. So what's happening is that usually you have protein that will interact with DNA first. They will recognize a bit of DNA that is already available because of some things that are unknown. So these transcription factors, they are called, these proteins will interact with DNA, and these will actually recruit protein that do these modifications. Okay? So when these proteins are recruited, they will modify the histone state. They will modify, these histone tails. And then these histone tails will then recruit the chromatin remodeling complex so that the gene that is being activated will be impacted. Okay? We don't understand what happens. Okay? Currently, what we think is happening and I give you, for example, something that is easy to understand. Is, for example, the glucocorticoid receptor. The glucocorticoid receptor, it's a DNA binding protein. Okay? It's a DNA binding protein, but it's a receptor. Okay? It's something that is not active. Okay? But in the cell, that has glucocorticoid receptor, when glucocorticoid comes in the blood, in the medium, whatever, this glucocorticoid receptor becomes activated. And this glucocorticoid receptor will interact with DNA. For example, here. Okay. This receptor has many activities. For example, because of its shape, it will recruit other proteins at this location. And among these proteins, there will be histone acetylase, histone methylase, histone kinase. They will all go there because they will interact strongly with this glucocorticoid receptor, so that the local chromatin will be decondensed, and this chromatin remodeling complex will then come and unpack the whole thing. But still, how the glucocorticoid can access the DNA that is packed into these chromatin fibers, we don't know. All these things is organized, and usually we think that the first thing that happens is a small transcription factor like the glucocorticoid receptor. They are usually quite small, and they can interact with DNA even though the DNA is packed in chromatin fibers. But then this very small protein can recruit other big proteins to completely unpack chromatin and start gene expression. But there are many, many things we don't understand. Okay? There are many things that doesn't make sense. Okay? Especially because inside these things, in addition to all these protein and DNA interaction, sometimes you also have RNA inside. Okay, like you have non-coding RNA, you have many things that interact with these, but what really happened at the molecular level, if we could take a look at this and see what happens when a gene is activated, see what happens when a gene is inactivated, really we can sort of imagine what happens, but we have no idea that is what is really happening. So, okay, maybe we think maybe where this linker, where this link is located is really important and different in every sense. We really have no idea. It's all conjectures. Okay? But all that we know, it's very dynamic, and there are many proteins that can regulate how chromatin is configured. Okay? So let's just remember that. Interphase chromosomes contain both condensed and more extended forms of chromatin. And this is what I told you before. So you have here a theoretical chromosome where you have the two ends, the telomeric end. You can see this is heterochromatin because usually at the end you have no genes. It doesn't need to be accessed. Okay? So this is heterochromatin, very stable, compacted form of chromatin. And centromere is also made of heterochromatin. But in between, you have various levels of uncondensed chromatin called euchromatin. But as you can see, there are different levels of compaction. Okay? It's not always the same. And some region of each chromosome can either be in heterochromatin or in euchromatin. And you can imagine in different cells, the pattern will be completely different. Okay? Because maybe in this cell, where the genes that are in this are not needed, so this is why it's condensed into heterochromatin. But in a different type of cells, maybe the genes that are encoded by this DNA will be needed, and then in this case, it will be condensed into euchromatin. This is very important because there is a disease. It's one form of anemia that is because of not normal compaction level. So there is one thing that happens is that you imagine a region of heterochromatin. Usually, these regions have specific tail modification. Okay? So you have histone tail modification that will promote the condensation of nearby chromatin into heterochromatin. And it will proceed until you have a kind of a, that's what they say here, they have a barrier DNA sequence. It's sometimes called isolating DNA sequence. It's a DNA sequence, a specific DNA sequence that will tell all this machinery, okay, you have to stop because from now on we are having genes and you don't want to compact this chromatin into heterochromatin. We need these genes. And so this heterochromatin will progress until this barrier DNA. And there is one disease, one form of anemia, as I was saying, in which there is a mutation in one of these barrier DNA. And unfortunately, the first gene after this barrier DNA is beta globin. And as you know, beta globin is necessary for hemoglobin formation. These people, they produce very low level of hemoglobin, so therefore they suffer from anemia. So the beta globin gene is okay. There is no mutation in the beta globin gene, but they have a mutation in one of these DNA sequences that stops the progression of heterochromatin formation. And so instead of stopping there, heterochromatin will continue and will absorb the beta globin gene. And the beta globin gene will still be a little bit transcribed, but very, very low amount. So therefore, this results in anemia. And probably one of the most amazing feat of heterochromatin formation and chromatin regulation is what happens in female cells. We are all told that females have two X chromosomes. Okay? Males, females, we have also human are the same. Human, female. They actually have two X chromosomes. We have XY chromosome. But actually, in each of female cells, only one X chromosome is active. Okay? So you imagine a female cell in the embryo. So you have the chromosome X that is inherited from the father, and you have the chromosome X that is inherited from the mother. Okay? Paternal X and maternal X. Then the ring development, they will be random in activation of one of the X chromosome. So for example, in one lineage, the paternal X chromosome will be completely condensed into heterochromatin. So the genes that are encoded by the paternal X chromosome will be completely silent. But in another lineage, this is random, okay? In another lineage, then this is the maternal X chromosome that will be condensed in chromatin. And from the first cell of that lineage to the end, and maybe muscle cell or whatever, always the maternal chromosome will be condensed to heterochromatin. So this is a form of cell memory. These cells remember that this first cell had its maternal X chromosome condensed into heterochromatin. And these cells remember that it was the paternal X chromosome that got condensed into heterochromatin. This happens, and we sort of understand now how it happens. It's actually via interaction of DNA with RNA. So I told you RNA normally, they code for proteins. Genes code for proteins. But sometimes you have RNAs that do not code for proteins. For example, ribosomal RNA. But in this case, it's called a long non-coding RNA. And what it does is that it's encoded by a chromosome, and it is only expressed in female cells. And when it is expressed, it will actually associate with the DNA of one of the X chromosomes, and from the first association, this will actually trigger condensation into heterochromatin. Now, this is a bit too much detail, but it's one of the very recent discoveries, and the long non-coding RNA that actually trigger the condensation of one of the X chromosomes in female cell is called XIST. And it's extremely long, maybe 15 kilobase RNA that its function is only to interact with DNA and condense locally. So anyway, this is what happens. So please remember these things, that it's very important for development sometimes to condense chromatin into heterochromatin, as happens in development of female cells. And that's the end of the chapter. Do you have any questions? Do you have any questions?
Comment.
Yes.
In another lecture,
Yeah.
a teacher said 30 nanometer fibers of chromosome
Mm-hmm.
is not exist in vivo.
Mm-hmm.
10 nanometer fiber, 11 nanometer fiber with a protein called cohesin or condensin is actually it actually exists.
So what the teacher said is that this is the form that exists.
Yes.
This form will sometime will exist where maybe you need to have higher access for a location of the genome that is heavily transcribed.
Heterochromatin is more condensed than
This is not heterochromatin.
oh
This is not heterochromatin. This is just a chromatin fiber. Heterochromatin is more these kind of things. Okay, it's when these chromatin fibers are even more packed, then this becomes heterochromatin.
okay, let's discuss later.
Yes. Okay, if there are no questions, this is the end of the lectures. Thank you very much.
Thank you.
See you on Saturday. Bye bye.
Saturday. Wow.
Thank you.
Bye bye.
Thank you.
Bye bye. Okay, so these chromatin fiber really do occur in vivo. They really occurs in vivo, but not where genes need to be activated. If you have genes that are transcribed or genes that needs to, at some point, need to be expressed, then it will be either chromatin, euchromatin, or heterochromatin. Okay? So,
Not where the gene is transcribed.
like, where genes are transcribed, you are right. So what you have, it's either, like, it's a little bit an intermediate between the beads on the string and the chromatin fiber. So this is more something like this. I didn't show it here, but this is something like this. So not as packed as a chromatin fiber, but not as unpacked as this. Yes. these beads on the string, these very long beads on the strings, really happen when you sort of do chemical treatment to the histone. Okay, but this is only observable when you do some experiments. Okay, maybe add salt or whatever. But the chromatin organization in active genes, okay, active genes is a little bit different. Maybe it's how could I draw? I'm not sure about the dimension, but a little bit more packed than the beads on the string, okay? Not chromatin fiber, but not beads on the string. A kind of intermediate where you have some proteins that interact with the chromatin to keep it in a form that is easily remodelable. And then there will be two forms of this one. Maybe this one is a euchromatin and is actively transcribed. And then maybe a little bit more packed will be heterochromatin. Okay?
Heterochromatin is heterochromatin straight.
So that's the thing, is that the word heterochromatin is very flexible, and then sometimes, even if this is even more packed, it's still called heterochromatin. I don't think there are currently terms like heterochromatin 1, 2, 3, 4, 5. I don't think this exists now. But as you can see from this picture here, I think maybe this is not so bad. This one here, this slide maybe is good for this discussion. No, you can see this one here. This slide is not bad. As you can see here, there are different levels of heterochromatin, and there are different levels of euchromatin. So exactly what is the dimension of chromatin and DNA is extremely variable. It's extremely variable. So different levels of euchromatin, but I would say that in vivo, when there is a region of DNA that you do not need, I think it forms a chromatin fiber like this.
Where you don't.
Maybe the most condensed form of heterochromatin, I think it's like this. いや、全然いらない場所。Which kind of makes sense, right? And then when you start to unpack into something that is a little bit more packed than this one, then this is a region where the cell needs a lot of access. But there are still many things, and you can probably get this is a field that also changes a lot. Heterochromatin, euchromatin review. This is sometimes there are many things. There are many reviews about heterochromatin. can check. I have another book which is the molecular biology of the cell. I give more details as to different levels of electrochromatin. There are some rules. That's what they say here. I think this is somewhere all for the stone. So then this one is experimentally unpacked. These electron micrographs show a length of chromatin fiber has been experimentally unpacked. And then, but it would be nice to actually find although long strings of nucleosomes form on most chromosomal DNA, chromatin in the living cell rarely adopts the extended bit on the string form seen in figure 20b. So the real form of accessible chromatin is maybe a little bit more like, not like this, but an intermediate form between the two. So no, this is really a fascinating thing, how all these things happen. Yeah, and I see here there are people studying what is the interaction between the machinery that repairs DNA and these chromatin fibers. Because if you want to repair DNA, you also need to do a lot of things, right? You need some time to break DNA, and you need to. It's very, very, very complex. And what happens between heterochromatin and DNA when you repair? Like for example here, repair of DNA double-stranded strand breaks in heterochromatin.
Are histone stable?
What do you mean?
I heard that DNA is very stable, so computer scientists want to use DNA as a storage.
Yes. That's fascinating as well.
But proteins usually degrade quickly.
Yes.
So is histone renewed frequently? Or
That's an excellent question. What is the half-life of histone? I have no idea. I really have no idea. How long the histone remain? Something that I have read is that when the cell divides, each daughter cell gets half of the histone.
half.
So half of the histone, and then the next half is newly synthesized. So, but what is the lifetime of histone? And is the half-life of each subunit the same? I have no idea. I really have no idea. It's potentially regulated as well. I can imagine. But we can check. We can check. Like maybe there is something like histone half-life. Where I cannot write histone.
Where is the gene which called histone?
I'm not sure which chromosome it is, but it's not one gene that codes for all of the subunits. It's different genes. But what do you mean where it is? Which chromosome or?
Where?
Okay.
It feels like a chicken or egg problem.
You mean the gene that makes histone has histone. So which we can check, look, for example, this one, histone H3, certainly we can find in the genes. Like, for example, this one. Of course, this is in Saccharomyces cerevisiae, but there is the gene HHT1, which is the gene for histone. And it's in the chromosome number 14. And this is the Homo sapiens gene. Histone cluster 1H3 family member B. Histone 2H3 family member A. So is it a big gene? So these are all histone. And this is funny, this has no intron. There is no intron. We can see the gene structure here. It's a very simple gene without intron. So no splicing, whatever. So that's kind of interesting. And they have no polyethylene. No polyethylene. So these are very strange genes. Transcripts from this gene lack polyethylene. Instead, they contain the palindromic termination element. This gene is found in the large histone gene cluster. Apparently, there is a gene cluster on chromosome 6. So it's a cluster. Oh, yes. Look. This cluster in the chromosome 6 in humans has many histone genes. So they are all, which is very nice, because if they are all grouped, then you can make sure that this whole segment is always available, or something, so the cell can always make it. And maybe the fact that DNA has no polyethylene, the RNA has no polyethylene, means that maybe the RNA must be very stable. The RNA must, because polyethylene is something that also regulates the half-life of RNA. So if this RNA has no polyethylene, then maybe it's a very stable RNA that can always make it.
What factors make RNA stable? I know that polyethylene or
it could be, but
no telomere, not RNA, not RNA. So they are mainly in RNA.
It could just be
What you see in books is the RNA is like this, right? You have the cap and you have the polyethylene. But actually, it's much more complex than that. What's happening is that you have the cap, and then you have a big complex of protein that interact with the cap, and you have a big complex of protein that interact with the polyethylene, and actually, they interact together. And it's many proteins, and it's maybe, and this is the 3' UTR, and there are proteins that also interact with the 3' UTR, so it's all sequence specific. And then among these proteins, some of them will actually regulate the RNA stability. So it really depends. They are cap binding protein and poly-A binding protein that will be the same for all transcripts. But the proteins that interact with the 3' UTR will be sequence specific, so they will be specific for each transcript. And depending on which protein can interact with the 3' UTR, the 5' UTR as well, they will regulate how quickly, for example, the polyethylene is removed. Because usually when you remove the polyethylene, then this is a sign that the RNA has already degraded. Okay? So the proteins that interact in a sequence-specific manner will actually regulate how quickly the RNA is degraded. And usually each RNA has two degradation points. Either you can degrade RNA from the poly-A, or you can degrade RNA from the methylation, the methyl cap. Okay? But first,
introduce the
First, you must remove the cap. You must remove the cap and digest
I
RNA.
know where it was. I knew it was happening, but I didn't know where it was.
Well, essentially, the key to remember is degradation is really specific for each RNA. Each RNA has a different half-life.
Is it here?
And what really regulates the half-life of RNA is which protein interacts with the 5' UTR and which protein interacts with the 3' in the sequence.
Is that the same this Saturday?
These proteins will either affect
Same this Saturday.
cap-dependent RNA degradation or poly-A-dependent RNA degradation. So there are many things. And yeah, there are many proteins that degrade RNA. But how they are regulated, what is their function, and so
I don't really speak that much.
and they are like even 5', 3' RNAs.
Actually,
So it's RNAs that degrades from there. There are actually two of them. There is XRN1 and XRN2. And it seems that one is in the nucleus, the other one is in the cytoplasm. The question that you have, how is the half-life RNA regulated, is an extremely complex question. There are people who are doing big work to understand how it works. It's very important. It's very important. It's a good question.
It seems there is very room for research.
Yes, exactly. This is one of the most intense research now. It's how RNA half-life is regulated and how gene expression is regulated. That's very important. Yes, indeed.